Hello everyone, my name is Rahul. In the previous video, we had discussed about nucleus. In this video, we are going to discuss about chromosomes. Now, during the time of cell division, the chromatin material gets condensed to form chromosomes. Thus, chromosomes are condensed form of chromatin. Chromosomes, they are not visible during interphase, but during different stages of cell division, they are visible. Chromosomes, they can be best studied at metaphase, during metaphase they are at the thickest and the smallest size. That is why it is very easy to study them. Also, they are arranged in the middle of the cell, which makes the study further easier. The number of chromosomes in a gamete is called as the number of chromosomes in a gamete is called as haploid number, which is represented by n, or we can say that a complete set of chromosomes inherited as a unit from a single parent represents the haploid number of chromosomes. All the chromosomes in a cell in their entirety are referred to as genome. Different species have different number of chromosomes, and the number of chromosomes, they are specific for each species. A single human cell has around 2 meters of DNA, which is distributed among 23 pairs, or in total of 46 chromosomes. Now, talking about the classification or the types of chromosomes based on position of centromere. The constriction which is present in the middle of the chromosome, it is known as a centromere, and based on the position of centromere we have different types of chromosomes. First is a telocentric chromosome, when centromere is terminal. And located at the tip of chromosome. Acrocentric is when the centrosome is subterminal, that is not present at the tip, rather it is located near the tip. Whereas a telocentric chromosome is a one where centromere is located at the tip. Metacentric chromosome is where centromere is located at the center of chromosome. In a submetacentric chromosome, the centromere is located near the center. It is located near the center. Now the ratio of long arm to the short arm of chromosome, it is called as arm ratio. It is the ratio of long arm, which is represented by P, to the short arm, which is represented by Q. Now this is a telocentric, this is acrocentric, this is submetacentric, and this chromosome is a metacentric chromosome, where it is the centromere is located at the center. Now karyotype. Karyotype refers to study of external morphology of all the chromosomes in a cell, which is specific for each species of living organisms. Karyotype, it is generally studied in metaphase. It includes number, relative size, as well as position of centromere, length of arms, secondary constrictions, and bending patterns. So all the details about chromosomes, they are studied in a karyotype. Ideogram, it is a diagrammatic representation of karyotype. In a ideogram, chromosomes, they are arranged in decreasing order of size, and sex chromosomes are placed in last. Ideogram, it is also specific for each species. By karyotyping and for performing an ideogram, we can determine if there are any genetic abnormalities in an individual. If there is any loss or gain of chromosomes, this can be detected by a karyotype, and this is useful in the diagnosis of chromosomal disorders. Now talking about the structure of a chromosome. There is presence of chromatid. Chromatid refers to two cylindrical arms joined at centromere. So these chromatids are known as sister chromatids. They are joined together by a common centromere. A chromosome may have a single chromatid or it may have two chromatids. Each chromatid consists of a single long thread of DNA. Each chromatid consists of single long thread of DNA associated with histone proteins. which help in the packaging of DNA. Non-histone proteins and RNA are also present. And RNA are also present. Now, talking about the centromere, it is the connection between two sister chromatids. Both chromatids of a chromosome are joined or connected by a structure called as centromere. At centromere, two protein discs are present which are called as kinetochores. Contains two protein discs. These are called as kinetochores. They constitute the actual size of attachment. They constitute actual attachment site of sister chromatids. At the region of centromere, the chromosome is comparatively narrower than remaining part of chromosome, thus it is known as primary constriction. At centromere, chromosome narrowing is seen. This is known as primary constriction. Now we have something called as a secondary constriction. These constrictions, they occur in certain chromosomes. They are non-staining. Also, they are found at a constant location. They are also known as NOR, nucleolar organizer region, also called as nucleolar organizer region, NOR. After secondary constriction, we have the presence of satellite. Satellite means it is a part of chromosome. After NOR or secondary constriction. Next is telomere. Telomere refers to the polar ends of chromosomes. These polar ends, they prevent fusion of one chromosome to another chromosome. These are rich in guanine. Now we also have an enzyme known as telomerase. This enzyme, it synthesizes telomeres and keeps the chromosomal size constant. This is a ribonucleoprotein. These telomeres, they shorten with aging and they do not disappear or shorten in cancer cells. So, with age, it is normal for these telomeres to get shortened, but in cancer cells, this property is lost, and thus cancer cells they are able to survive for a long time as compared to normal chromosomes, which usually disappear due to the absence of these telomeres with aging. Now, there are certain special types of chromosomes. First is known as a polytene chromosome. A polytene chromosome, it is referred to as polytene because number of chromatids are very high. Swollen areas present at some places in polytene chromosomes are called as Balbiani rings. It contains swollen portions called as Balbiani rings or puffs. These puffs help in synthesis of RNA and proteins. Synthesis of RNA and proteins. These are concerned with metamorphosis and molting process of insect larvae. Second is the presence of lamp brush chromosomes. These lamp brush chromosomes they were discovered by Fleming and Racket. From oocytes of vertebrates. From oocytes of vertebrates. Mainly amphibia during diplotene stage. These are associated with lamp brush like appearance, hence they are known as lamp brush chromosomes. They contain DNA and RNA as well as proteins. They are concerned with vitellogenesis. They are concerned with vitellogenesis, that is formation of yolk. So with this we finish off with our discussion on chromosomes. See you in next video.